So for those who don't know me, I'm leader of the National Deuteration Facility at ANSTO. As Zoe mentioned, I'm currently chairing the board of the Deuteration Network, which I'm going to talk about today. In talks, similar talks like this, I like to start with a little story just to show you the impact of deuteration on a personal level. Some of you might have heard that story before. But really, about 18 years ago, I was doing my PhD at ANSTO. We were studying CH activation, how to break a CH bond with a catalyst, and we were studying these systems using NMR, low temperature NMR. And we got really nice results. And I remember halfway or towards the end of my PhD, still a year left, and I was writing the papers, like any other PhD students, trying to get this paper out. I remember my supervisor, Graham Ball, came to the lab and said, Tamim, look, if you want people to believe us and you want to publish the work in the high impact journals, we need to do the experiments with 2 more deuterated molecules. And I said, oh, that's fine, let's buy them. And he said, no, they are not available, you need to make them. And I said, okay, I'll make them, and it will take me a month or so. It took me 12 months to do the deuterated molecules, and that's because there were no commercial source for those molecules. There were no deuteration facilities available to do the chemicals that we wanted. So it's always I always find it very motivating to be in that community now, trying to help researchers and scientists and PhD students to access deuteration facilities. And today I'm going to talk about the Deuteration Network, which is the consortium or a group of deuteration facilities and laboratories across the globe. We're trying to actually get together and try to provide these services to the users. Some of us are user access facilities, and some of us are just laboratories that they do their own deuteration research. So this is the outline. Why I'm going to talk about today, why deuteration, why we need deuteration for the different techniques, a brief history about DeuNet, and then the refreshed DeuNet. So you can see there is a difference between small n and big N. That's how we move from old DeuNet, which I'll tell you about in a minute, to the new one and its vision and mission. Why do we need deuteration facilities? So questions that we always need to actually articulate upward to the managers, why, and our management, why we need to have. It's a cost effectiveness. I'm going to talk about this, and then I'll talk about the impact on science, showing some case studies from the different facilities. And if I have a little bit of time, I will show you some of the results from the user survey summary that I think some of you might have, as a users, have filled this survey last year. So, deuteration as a characterization tool. We all know deuteration is quite important for neutrons, because neutrons interact with with hydrogen differently than the deuterium. Different scattering length between hydrogen and deuterium makes neutron and deuteration a very powerful combination. And you can do contrast matching and so on. NMR, deuterium is a spin half silent or silent in proton NMR, so you can benefit from having clearer or cleaner spectra, but also you can do lots of other experiments with deuterium because it has a different positive effect. Infrared, the CH bond and the CD bond resonate at quite different wave numbers. And also lately, pharmaceutical companies been using this kinetic isotope effect, which is when you remove hydrogen and put deuterium, you make the bonds slightly stronger. You can trick enzymes by doing so. So you can make deuterated drugs, for instance, more stable. You can use the same technique or approach to stabilize other molecules, not necessarily drugs, that are susceptible to oxidation, for instance. So I'll talk a little bit about this too. So a nice illustration of how neutron and deuteration works. This is just an optical example of refractive index difference between the beads and the oil there. If you match the refractive index, you can actually you can mask that beads. So you can do highlight and de-highlight stuff. This is in the characterization field. If you deuterate molecules, you can change their physical properties to the point that they can actually new molecules that you can use. Just an example, deuterated ice is different than ice made from normal water. So if you make ice from heavy water, they will sink in light water, while normal ice will float. So you can see there is a difference in its physical properties. This is why here the density difference what makes the difference. So deuteration, to understand complex structures, we need to, in systems like this is lipid nanoparticles, this is the mRNA, LNP. If you want to understand this assembly of these lipids, what you need is basically to do, you can do light scattering, you can do X-rays, but if you want to understand the different components, you need to deuterate and do SANS, neutron scattering. These lipid nanoparticles are made from different kinds of lipids and sterols, cholesterol. So it's important to be able to provide those deuterated molecules to users who are interested in doing SANS on this type of systems. Reflectometry, neutron reflectometry, you can study still in the soft matter, where you can do devices. This is an OLED device, organic light emitting diodes. They're made from thin organic films sandwiched between two electrodes. And if you want to understand the diffusion properties, this is one of the issues with OLEDs. When they got hot, when they become hot during operation, or if they are sitting in the sun, they lose efficiencies. And that's because the thin layers they migrate to one another. Those organic layers. And in order to understand them, you need to discern between those organic layers. The only way to do this is to deuterate layers, or alternatively protonate them and do neutron reflectometry. If you do X-rays, you don't see difference, but if you do neutrons, you'll be able to see the difference between the three. This is the work we did with Paul Byrne and Ian Gentle, where they studied the diffusion and what causes the diffusion, the structure of the molecules, for instance. Partially deuterated proteins, quite important also for contrast for neutron scattering. You can understand interaction of proteins with lipid membranes, understanding bacteria function, protein behavior, food products. It's also used neutrons in food. You also need deuteration. In crystallography is another technique that benefits from deuteration. You can make smaller crystals if you deuterate proteins, for instance, shorter diffraction time, and you can have a weaker neutron source to get structures. So this is in terms of characterization, but if we move to the other part, which is it's a characterization tool, but it's also can be used as a for their own interesting properties, the deuterated molecules. In the drug fields, as I explained before, deuterated drugs. This is an FDA-approved molecule since 2017. There is a pipeline of deuterated drugs that's going to be in the market in the next few years. I think it's already 2 or 3 in the market now, but this is the first one that was approved. If you make those positions deuterated, you can actually change the half-life time here. It's about twice as long as the undeuerated or unlabeled ones. So you can see that you can improve the pharmacokinetics. You can reduce toxicity in certain drugs, and also you can modulate epimerization. Certain drugs that we take, sometimes they do epimerize in the body, and because of this, it can actually invert to a molecule that becomes toxic. You don't want that to happen before it gets metabolized. So you can, if you deuterate certain positions, you can slow down this epimerization process. So a brief history about DeuNet with a small n. This is a 4-year European Union project from 2015 to 2020, led by Hannah Buckland. Nick, you can see that this is the photo, I think, about around the 2015-16. So the funding was to, and ANSTO, Peter, some of you might know Peter, he retired now, he was my boss. We were an observer on this initiative. So the goal was to benefit from method and expertise and resources from a group of a number of facilities in Europe, form a cost-effective platforms, and also to include universities and international partners, and offer user access and coordinate access to all European users. So that was really a good start for the DeuNet. That's when it started. Then more people joined later on, but I'll tell you a bit more about the achievements that were achieved. From this grant or the establishment of this led to the basically the chemical deuteration laboratory that's in ESS. So that's actually how it was established from this grant. The European user survey was conducted in 2017, and SDFC ISIS in UK started to provide the deuterated molecules to European users, and development of methods for lipid deuteration, also in separation from cell culture at ILL. Also, there were collaboration between ULIC and ESS. ULIC started also make their deuteration services in polymer chemistry. And then there were more participation from in broader infrastructure like LANS and others. And then several new international members start to join the DeuNet. And I'll show you a map in a minute. But what's currently on, so all these funds done, I think that we run out of this, so it's actually running now by ESS, the deuteration, the DMAX. And it's actually part of, well, there were more funding through Brightness, which provided work package, which is a collaboration between ESS and SDFC. And currently, DeuNet is represented by John Webster from ISIS on LANS, basically, just as an example of a successful project and how it was implemented and the networking that happened later on. So from there, the deuteration facilities grew significantly. You can see, number in Japan, 4 of them, labs, China, in Europe, a couple of in the US, and 1 in Australia, which is the NDF. So we decided that well, Hannah couldn't just carry on with the DeuNet responsibility, so we had the decision that we had to make, which let's actually form a board. So there is now a board for the Deuteration Network. Hannah is still involved as an executive board member, and we have a board members here from all the different facilities and laboratories. Pejun, Zvi from NIST, Wolfgang is with us, from LP3, Hiro from JPAR, Hiro O'Neill from Ornell, Hiroshi Naka from Kyoto, Jürgen from JCNS, Kunlung from Ornell, and Howard Wang from China, Masaki from Kids from Japan, and Sajiki, Professor Sajiki from Lifo Thomas University, and the D-Lab, ILL. I think Trevor had a bit of contribution at the beginning before he became so busy, and then, but he still.
I think we sort of started in the early 2000s. That's where it all started for biology.
Yeah, that's right. Yes. And we have also a communication group or subgroup that is working on our outreach activities, which I'll show you. So the DeuNet vision and mission is an international network of deuteration facilities and laboratories, which aims to facilitate access to deuteration services and customized deuterium labeling of molecules and biomolecules for use in neutron research and other characterization techniques. So we aim to become a central hub for access to deuteration research around the world, deuteration information, and outcomes to highlight outcomes. So we aim to promote collaboration between the facilities. So one of our aims, or biggest aim, is to make sure that there is no duplications and steer away from competition. We'd like to become a body that can serve the users for their neutrons. We don't want to compete on capabilities. We would like to complement our capacity, capabilities to provide as much as we can deuterated molecules to users, whether it's for neutron or other applications. Increase the visibility of its members to researchers and research facilities that benefit from deuteration science, and facilitate the development of new methods of for deuteration, and facilitate communication between each of the DeuNet members and their collaborators through regular meetings and workshops. So, we hold the deuteration or DeuNet facilities meeting. This is the last one that we did in November 2022. I had that online, and also we have user facility or user meetings, and we were lucky this time to have an in-person tagged to European conference, ECNS in Germany, in Munich, early this year. So we had a DeuNet micro symposium that we run. So we had a lot of the deuteration people scattered in this crowd, and we gave basically talks, and we had a lot of good networking with our users. So we'd like to continue doing this on an annual basis. It's really good to be as part of a neutron or international conference, so that we can have the maximum exposure that we can get. JPark runs their deuteration workshop every year, nearly every year. This is from 2017, when Anna Leong from ESS, myself, were invited to the deuteration JPark or the JPark deuteration workshop in Japan. Kunlong from Ornell, myself also in 2018, and lately in 2023, earlier this year, we had a bigger workshop. Unfortunately, Zoe couldn't join us, but it was really good. So you can see they're actually getting bigger, the community there in Japan, new faces. And what was interesting is that we had a lot of ECRs, young, early career and young students joining this workshop. I believe that they're doing really well in terms of training the next generation deuteration. The amount of students that they get from the different universities, the amount of work they're doing is great. So, I think we should, we should watch the space for the Japanese being the next deuteration chemist, which is great to have. The next year JPark deuteration workshop would be mid-2024. Professor Sajiki, who was my host during this visit to Japan, from Gifu Pharmaceutical University, he's working on a grant. Hopefully, we'll get a grant that we will be able to run an international workshop, deuteration workshop next year, mid next year, hopefully somewhere in Japan. So just keep that in your calendar. So web page. So the DeuNet now has a new web page, deuteration.org. This is just a video slide of how it looks. You can scroll, through this. You see a map. This is the different partners, logos, and I'll wait for this to nearly finish, and then I'll scroll up. This is the map showing the different deuteration facilities, kind of like what's the nearest destination, what's your nearest deuteration facility that you can access. And if you hover on them, you can see their names. And if you click on any of them, it will take you to a different website, not the website of the facility, but a different website. We're trying to streamline this, which then you'll be able to compare and see which the facility is, the capability of each of the facilities, and what they are unique for, basically. So this is the NDF and how you can access them, whether it's a user program, whether there is any a portal, submission, due dates, who to contact, and also their website. So you can click on their website and get to there to see more information. Scrolling back, going back to, so I encourage you to go and navigate. This is in work in progress, so we are actually having also Twitter account tagged or in that web page that we will upload all the nice stories or the good stories and impact stories on the web page. This is another view for the members. If you go to, for instance, the DMAX here, you can see that their capabilities, their specialties, and how to access them, and who is to contact in case of any question. So next slide. So deuteration.org is your website to go to. Contact at deuteration.org. You'll be able to send any of your inquiries. If you have any molecules that you'd like to deuterate, you can send this. News and stories, you'll find them there soon. Publication, basically here to show the impact of deuteration. So similar of what I'm going to show. And also we have a chat channel for members only, where we can actually collaborate between us. Now some of us might be able to do a precursor that can be handed to another facility, where we can actually take that to the final product. I think that's Trevor had suggested this. I think it's going to be a little bit challenging, but I think we are aiming to go to implement this channel to keep the communication between different facilities. And there is a way we're working on how to get to you for any inquiries, get back to you on any inquiries that you do. So there's a mechanism there. I'm not going to go into it now. On social media, if you have used any of the deuteration or deuterated molecules from any of the DeuNet members, you can actually tag or hashtag, whatever they call it, on Twitter at deuteration, and that would show on us. And it's important to have this, because it will keep our funders happy that they are, this is the impact of science. Then, help us, we can help you back. And it's quite important to have us involved in your publication. We'll be able to provide you with the deuterated methods, or at least an acknowledgement if that's through negotiation. But that's actually quite important KPI for all the deuteration facilities. So why do we need deuteration facilities? I'll talk about the impact, the science impact later on, but I'll be focusing on the cost and effectiveness. Really customizing molecules to fit the user needs is more productive and cost effective. In the next few slides, I'll try to convince you in doing this, in this. So Carl is one of our talented synthetic chemists at the NDF. He comes from an industry background, so he's done those slides where he showed the difference of costs involved in making the deuterated molecules. Anyway, here benzyl, chloride, bromide, iodide, different versions. If you want to buy them in their protonated form, on this here, CH2, so 4 cents, 18 cents, 31 cents. You can see this is the trend. If you start to put deuterium, yeah, it's no secret, it gets significantly expensive. But here it gets, it's more expensive from 4 cents a gram to 18 cents a gram. But if you put deuterium on the bromide version, it actually becomes less expensive. So, and for the iodine, you don't have any price for it. So, who knows how much they're going to charge if you want to buy it from commercial source. So it shows that the deuterated molecules that you can get from commercial sources are driven by supply and demand, and not necessarily by how expensive it is. So, which makes me say that talk to us, because we can provide deuterated molecules a lot cheaper than what you think or the commercial people think that it can cost. So this is a nice example. Another nice example. This is deuterated fatty acids. We make the deuterated fatty acids by hydrothermal reactions. So you start with the fatty acid, you put it in a power reactor, high temperature and pressure, in heavy water, and with one cycle you cleave all the CHs, put deuterium instead. You have some hydrogens left, so not all of the hydrogens will be replaced with deuterium. But if you do one more cycle, you will getting towards the higher deuterium enrichment. But the cost versus the deuterium level is quite significantly different. So for the first cycle here, this is a price per 100 milligram. It's about $20 Australian dollar per 100 milligram for the first cycle of the fatty acid, where it will give you about 90% deuteration. If you put in another cycle, it will be about $38 per 100 milligram, but the increase in the deuteration will be from 90 to probably 96, 97. A third cycle will be significantly more expensive, about $60 a 100 milligram, and it's only an incremental increase between the 97 to the 98 or 99. So you can see a linear increase in the cost versus a very exponential flat increase or incremental increase in the deuteration level. And so, and when you go and buy deuterated molecules from commercial sources, they will sell you the ones at the highest deuterium level, which will cost you arms and legs, because that's how they made it. This is the demand for it, or this is how they stock it. But really, your 95% is good enough for the experiment, and it's actually significantly less expensive. So less expensive for us to make, which means that it's still efficient to make it and to provide it to you at a larger scale, for instance. So yeah, talk to us. This is oleic acid. Oleic acid is a quite popular or very important fatty acid that we make, others make also. We'll be making this on really large scale, and also are we able to provide it to other facilities to do their lipids. I'll just focus here on this slide here, showing the prices. So from a commercial source, you'll be able to get oleic acid with 2 deuterium only at $30 US dollars here per milligram. Put 17 deuterium, which is half of the chain, it's $82 per 100 milligram. From the same commercial source, you deuterate the whole molecule, so 33 deuteriums, 100% deuterated, 31, so it actually goes cheaper, $31 per milligram. From the NDF, we've been making only 1 deuterium less, so it's 32 instead of 33, and it's an order of magnitude cheaper. So the difference between 94% deuteration and 100% deuteration, it's not a significant difference if you're using neutron scattering, but it is a significant difference in the cost. Lipids is another story. From a commercial source, this is a POPC lipid, which is one of the important lipids in our membranes. So you get different parts of the molecule that can be deuterated. From a commercial source, if you want to have non-deuterated molecules, $3.1. If you deuterate only one tail, the magnetic chain, it's 33. If you deuterate both chains, and that's from us because it's not commercially available from the NDF, it's a lot cheaper. It's about four times cheaper, right? And if you want to put also the head and the 2 tails, it's 48. So yeah, it is expensive, but it's significantly less expensive than a commercial source where they deuterate the whole molecule. And it's an order of magnitude different. So yeah, again, talk to us to be able to provide these things. Case studies. So what I'm going to show you here is just the impact of deuterated molecules that have been accessed from the different deuteration from the DeuNet members, and how they basically produced an output, a research paper, maybe a cover letter, a cover journal, and high impact journal. So some of the case studies are only from some members, not from everyone. I'm still in the process of collecting case studies from everyone. And it's also it's only one or two case studies, one or two molecules made by the facility. So I'll start with ANSTO NDF. This is a molecule that actually we made with ILL with Trevor. They made deuterated cholesterol. We made two cholesterol versions, I think, for this paper that was published in JACS and received the cover journal. The neutrons were done at I think an ILL and are actually at ANSTO, neutron reflectometry. Basically, this is understanding the mechanism of the spike protein, the SARS-CoV spike protein, how it infuses to the cell membrane, the host membrane, and what is the mechanism of penetration. So that's actually quite important to understand. It's quite important to deuterate the different components of the lipids, whether the model membrane or the different components of the lipid membrane, whether it's the lipid or the sterols, in order to do this neutron reflectometry. Another study from NDF, from ANSTO, where we had produced for an industry, Mitsui Chemicals from Japan, bucket loads, if you like, tens of grams of deuterated mineral oil. This is mineral oil is a solvent that they used in the manufacturing of polymer, and they were interested in studying the formation of the polymer in situ while using sands. They had two options, either to deuterate the solvent or deuterate the polymer. Now, if to deuterate the polymer, it's quite complex, and also you're not going to get the same polymer that you will have as a protonated version. So the idea was to deuterate the solvent, but the solvent is mineral oil, so something that's not commercially available. So we devised actually a nice way to make tens of grams of deuterated mineral oil that they used, and that received a journal cover also in polymer chemistry. Another study that used deuterated cholesterol that we've been making at the NDF at ANSTO, also to understand the assembly of the lipids in the LNPs, the lipid nanoparticles, and how what's the impact of binding the apolipoprotein to those lipid nanoparticles? What happens to the cholesterol? Also, in this case, you need to do the contrast variation and to be able to localize where the cholesterol assembled in those lipid nanoparticles. So this work was with Marit and with AstraZeneca, and also the neutrons were done in ILL, and also in collaboration with others, as you can see there. So some highlights also from DMAX. This is the work that they published recently, showing the output from the different capabilities that they have. So they do deuteration, chemical deuteration, biodeuteration, and also crystallography. So this, I guess, this is the crystallography story here, where they deuterated this amidase from an Antarctic polyextremophile to study basically the enzymatic mechanism using a neutron crystallography, and also they used ILL. They used it from ILL and also from MAX4. Another paper published recently also, where they were interested in studying the shape and the function of these eutectic solvents. These are the quaternary ammonium salts, ionic liquids. When you mix them together, they have a melting point lower than the individuals. So when you mix the mixture, it becomes lower melting point, less than 150 degrees. So they stay as a liquid. So it's actually quite an important type of solvent. To understand the micellar formation, they use sands. Of course, they needed deuteration. So they were deuterating they deuterated the alkyl chains of these solvent molecules. Another paper, also where they deuterated, I guess here, the lipids in order to study the interaction of this enzyme that binds to the protein in a lipid bilayer. This is, I forgot the function of this enzyme, but I think this is an important enzyme in the formation of amino acids to make our DNA. Funny, I think that's one of the enzymes that's important too when it binds to the protein in a lipid bilayer as a function of lipid composition. To understand that dynamics there, sands were used, or reflectometry were used in this case, and also the deuteration of the lipids. The other case study here from the news from Loon Protein Production Platform, LP3, with Wolfgang. So Sweden enters into an in-kind agreement with ESS. That's to strengthen the contribution of the Swedish science community with the ESS. Basically, they had a number of projects, 4 projects, contracts. One of them is the deuteration lab services that is part of the DMAX now, and it's led by Wolfgang Nick. He's the head of the LP3 and also the director of the protein production Sweden. So the idea is to support researchers to do life science experiment and also in the crystallization of molecules. So that's a really good value to add into the DMAX capability through the LP3. Some news from our friends in Germany, in Jülich. They are the experts in polymer deuteration. So anytime you need deuterated polymers, I think these they are the people to go to. This is a nice study that they use deuterated PEG molecules to basically study or resolve the structure of soft gels, nanogels using sands. And you can see this is the synthetic method that they can make really nice mono dispersed or narrow dispersed PEG molecules. MW of Mn nearly 1. Nice story from Oak Ridge Deuteration Facility, also recently published in Nature.com. Nice, well, first time study of protein encapsulated into MOFs, metal organic frameworks. So it was important to deuterate the protein. They actually used the GFP, green fluorescent protein, and they used I think neutron scattering, yes, sands, where they studied the conformation of protein inside those nanopores, and they found that it has a different conformation, changes that is observed, different than how the protein behaves in solution. So this is a quite important study because it is actually has an impact on application in biocatalysis or on drug delivery as a drug delivery vehicles for the protein. From JPAC from Japan, they do biodeuteration and recently started doing chemical deuteration. In the chemical deuteration, they focused on ionic liquids and well, in this case is as dangling groups of a polymer, hydrophobic, hydrophilic separation. And they use reflectometry to study the microphase separation. The study in the top was also published recently, or in 2019. Here they actually made deuterated whole cell debris because they want to make very much like a mimic the environment of a living cell, and they were interested in this in the studying the interaction of protein in such a crowded system or environment. From combinatorial deuteration station project, kids, they have they do biodeuteration, they do quantification of deuterium level, they do large scale protein production of deuteration, they have capability of recycling DTO also, but their specialty basically is the domain selective deuteration through ligations of proteins. So this is a study that been published in Angabante last year. So that's actually also a nice complementary capability that is available from this group. ILL in Grenoble in France. So since as probably most of you know, since January 2022, a new team of is managing the ILL deuteration facility. That's when Trevor moved to Leeds, to here. And now they actually opening a position for the head of deuteration biology chemistry of soft matter science support group. This is their website. So one of the highlights they did published a couple of years ago about deuterated fucose, a monosaccharide through an engineered E. coli. So that's a nice sugar to deuterate, but it enabled actually 2 or 3 more studies and 3 more publications there. And this study here they did deuterated the lectin from lectin is a sugar binding protein, and they did the crystallography. They co-crystallized deuterated lectin from insect pathogen, and also with the deuterated fucose. So you can see nice density map there, which is enabled by the deuterated components of the lectin and also the fucose. That was published 2021. They did the same thing with human pathogen from Pseudomonas, a very dangerous bacteria affecting lungs. Also studied the interaction and the hydrogen bonding between the sugar and the lectin made from the Pseudomonas. So it's actually quite important work. This is actually in collaboration with CERMA. The work here basically is important because it can design new glycomimetics against human pathogens, so in terms of therapies. And also this received, Lucas basically received an award for this work. He was doing his PhD, so during his PhD, he received it from a crystallography conference. ISIS deuteration facility. This is a nice story from, well, very recent story published in ACNS Nano 2023, I mean this year. They were interested in, with their users interested in studying the shape of the lipid nanoparticles upon protonation or acidification, how it changes, and it encapsulates plasmid DNA basically. And they were interested in how acidification can change the structure, the volume, the distribution of the lipid components of these lipid nanoparticles. And for this, they used a number of lipids. Some of them are, well, what they've done here is that they provided this MC3, is a quite important lipid used in LNPs for mRNA technology. Because of their synthetic skills, they were able to help their users by providing the protonated version of this lipid. Now, in the NDF, we just a couple of weeks ago, we had actually produced the deuterated version of this molecule. So there's a quite good demand from users to access this molecule in its deuterated form. So we publish also papers and get interest from editors. It's a journal cover only as methods rather than application. So this is a paper that we at the NDF published early or late last year. In advanced synthesis and catalysis, we described a way of making linoleic acid. It's a very lengthy steps, but we had a really nice, robust method describing it step by step. And the editors and the referees thought that this is actually quite important, so they offered us to be on the journal cover. So that was quite nice highlight. You can use deuteration not only for neutrons, and that's I think that's my next 10 minutes. You can use it in applications like nanosims, IR, mass spec. But nanosim is a secondary ion mass spectrometry imaging. where it can do tracing of isotope tracing. And in this case here, we provided deuterated cholesterol, where our users were interested in studying where the cholesterol locate or deposit in the intestine of animals. They used mice models here. They fed them with the deuterated cholesterol, and they looked at the distribution of the cholesterol in the microvillei of the intestine. That's where they actually most of the exogenous cholesterol goes into. So it's actually a nice study. So as I said, different applications for deuteration. Kinetic isotope effect is attracting a lot of interest lately. And that's why removing hydrogen, putting deuterium to make the bond stronger. Stability of material is basically the enemy for everybody, right? Like from aging, skincare products, they degrade when you sit in the sun. Electronics, they rust. Breakdown of molecules, enzymes, all of that, it suffers from stability or instability. And the common between all of them is basically oxygen. Oxygen, we need oxygen to live, but it also, it's behind what causes us actually to die. So, and that's because oxygen breaks bonds and insert itself and do oxidation. Enzyme does the same thing. Enzymes in our body, when any drug molecule, which tend to be, well, the molecule need to go to the target to do some effect, but in its journey through our liver, the enzymes start attacking it and degrades it. So you need sometimes to stabilize the drug a little bit more so it reaches the target. So that's another impact of instability. The DeuSwitch team from Kyoto University is another small but effective, very effective team working on deuterating drugs in Japan. They do catalytic deuteration, they do calculation, which is quite important. You still need to, it's good to do calculation before you start doing the deuteration, because it will tell you where to deuterate and where not to deuterate. In situ analysis, they do a lot of applications. So they have started deuterating drugs like this one here, published in Chemical Science Journal, losartan, potassium D2. They prove that this is actually more effective if you block the enzymatic degradation of this group by deuterating this site. And also they're applying the same technique on catalyst. So catalyst does the same thing. Catalyst acts to make, so for instance, this catalyst here is called a Maruka catalyst. It's a catalyst used to make dialkyl amino acids. If the catalytic cycle can be interrupted and the catalyst can get oxidized and it stops making its, or stops its function, and that's not good. And what they've done is they've reinforced where it can get cleaved or oxidized and put deuterium there, and they basically have a superior catalyst. So that's actually a really nice application using the deuterating drug approach, but on catalyst. So this is published lately last year. I was talking about enzymes and how it works. Also, metabolism of ethanol. This is another nice story. These guys are not part of DeuNet, but I like this case study of showing the importance of deuteration. Deutera beverages, I'll tell you the story in a minute, but the ethanol is not the bad guy for us. What's bad is the acetaldehyde. When we drink, the toxicity effect comes from the acetaldehyde, and then when this builds up, this is what makes us drowsy and toxic and whatever, and a hangover. There's another enzyme, aldehyde dehydrogenase, basically convert this to the acetic acid, which is actually good for our body. So the accumulation of this transition molecule is what's bad. So what they've done, their theory is if we can deuterate the alcohol, we can actually have heavy alcohol light on our head, kind of thing. So I'm not encouraging anyone to do this, but that's actually an interesting concept. Deuterating ethanol, you can actually slow down this kinetic reaction and keep the other one not impacted. So what we've done, at ANSTO, we've been doing also, as you can tell now, we've been doing deuteration for neutron, NMR, mass spec. Also, we were also doing some deuterated radio traces. So ANSTO is famous on with the radio traces. We do imaging, radio labeling. So radio tracer is a molecule that you basically put. It's a ligand that binds to a protein of a diseased organ. You do PET imaging, and you put a radionuclide on it. One of the molecules that we were interested in exploring is this PBR111, which actually it images the neuroinflammatory diseases. One drawback for this is this radioactive fluorine, when it passes through the liver, it gets cleaved at this site, and then it further cleaved to liberate only free fluorine. Free fluorine will love calcium and will bind to calcium everywhere it can get. So when you do a PET image, you will get signals from the bone as well as from the tumor. So that's not good. That was a case study. I mean, that's not the best molecule that we could have worked on, but we worked on it because it was handy and available for us. So we said, okay, we're going to stabilize this a little bit more. And we put deuterated molecule atoms here at this carbon and at this carbon. And we fed it to, or basically injected it to the rats, and we did PET imaging, and we showed that there is less bone uptake. So that means less fluorine is being cleaved in comparison to the protonated version. This work was published here a couple of years ago. But that was a clear demonstration of how that molecule can be stabilized by deuterating certain places in there. So this is another image showing that in this case, when it's protonated, you get signals from most of the bones, including the kidney, I think, or the spleen, somewhere there, where it actually resides. A lot of the fluorine sits. While it's deuterated, all or most of it gone to the tumor in the brain. So it's important to know where to deuterate, because we've also made deuterated other things like sunscreen, and that didn't work. We made the sunscreen even worse than what it is, so it degrades very quickly. So it's important to know where to deuterate. So we're calling it to D or not to D. So that's the slogan that we use now. And it's important to do this before you start doing it. So if you can do molecular calculation, like what the Japanese are doing, that's actually quite helpful to know whether there is a kinetic isotopic effect or there isn't. Right, so 2 minutes, I think I got to, and that would be my full hour. I'm going to go quickly through the DeuNet user survey summary that we received last year, towards the end of last year. We received 305 responses from a survey that we sent from all the Deuteration Network to the users. And most of those users come from Europe, which was surprising for us. But it shows how important the user base in Europe is quite big, and they're interested in at least filling that survey for us. But that was good. So 50% of the current users of deuterated materials responded that their research is very dependent or impossible without it. So that's actually a quite significant result that we observed. 83% they said it's dependent and above. So it's actually quite a good number. So you can see out of this selection, or that's part of the 306, there were many of them depend on deuterated molecules. So what's that about? Okay, 50% of the total survey respondents didn't know about the DeuNet, but and so it was an insightful information to know. We need to do more outreach, need to do more talks. So if they know more about us, hopefully we can help them more. So that's something that we learned. These are the type of molecules that this selection of surveys were interested in. Electrolytes, solvents, probably that's it's over because of the solvents. Maybe we should change that next time. But proteins still one of the highest ones. Sorry, polymers, lipids, detergents, aromatics, sugars, fatty acids, sterols. So, but it's a nice distribution. It shows us where the demand is coming, at least from this direction. SANS is the highest, as one can tell. SANS, another neutron, most commonly used experimental techniques. If we say to the users, what would you like if you have deuterated molecules? So we listed the question there, and we saw a spike in the NMR. It seems like there was a lot of people that they know if there is more deuterated molecules, they probably can use more NMR. So that was actually insightful for us also. The challenges, we're going to post this on our webpage. You can see, I'm just going to go to the last one. One more. I think I want to just end up with this one. This is an interesting one. So responses to a question regarding cost of production of deuterated materials. Many said that, yes, we are happy to provide funding or ask for funding through the funding agencies to be able to provide it to the deuteration facilities as a cost recovery model. We're not suggesting that it's going to happen, but at least we know that the users are aware of the value of these molecules. Some of them said, yeah, we're happy to contribute to the cost. And some of them said, no, but that's understandable. It's important, as I said, it's one of our KPIs, I guess, for everyone, is to show the impact of the facility to our management. And having the deuteration people or the person who did the deuteration for you on your website, on your publication is quite important for our existence, basically. So this question was saying, are you happy to put co-authorship? And the majority were saying yes. So that was quite useful information. At the end, I'd like to thank you all for your attention. I'd like to thank the DeuNet members and their associates. These are the board members, but a lot behind basically making the molecules and helping the users. And thanks for your attention. Thank you.